Hello, everyone. My name is Lori Hoagland, and I am an associate professor at Purdue University in the Department of Horticulture and Landscape Architecture, and I'm working with Petrus Langenhoven, on a project, a SARE-funded project, to deal with the problem of Verticillium wilt in mint crops. And so today I'm going to talk a little bit, a brief overview about what we're doing as part of this SARE-funded project around anaerobic soil disinfestation. I'm going to give you an update on the research that we've been doing up to this point, both in the greenhouse and field around ASD technology and Verticillium wilt. And then lastly, I'm going to talk about a few things that we're learning about the ecology of this pathogen and some ways we might be able to think about better managing it in the future as an alternative to ASD or as part of ASD, and just giving growers more options. And so we really want your feedback on this. So as many of you know, Verticillium wilt is a disease that attacks mint plants, causing really devastating losses and destroying entire fields. And that is caused by this little pathogen called Verticillium dahliae up here in the right-hand corner of the slide. And this pathogen, it can come into a mint field a number of ways, through infected transplants, infected equipment, what have you. But once it's in the field, it can get into the mint plant, it gets into the vascular system, blocks the plant's ability to transmit water and other nutrients through the plant. And so eventually what happens is the plant starts wilting off like this and eventually dies. And then during that process also the pathogen is producing long-lived spores called Microbial, which are theorized to be able to survive in soil for up to 14 years without a host, which makes it very challenging to deal with this pathogen once it's in your field. And so the traditional ways of dealing with the pathogen are to move to a new field, if you can, a clean field, which isn't always possible. And so instead you can apply broad-scale chemical fumigants. However, these are expensive. They're becoming more challenging because of new environmental regulations, because they can cause a lot of harm to the environment. And then lastly, it's important to understand that these broad-scale fumigants not only kill the bad organisms in soil like Verticillium dahliae, but also the good organisms in soil that are doing things like helping fight pathogens or cycling nutrients. So anaerobic soil disinfestation is kind of a newer approach and is thought to be a very successful non-chemical approach to control soil-borne pathogens like Verticillium dahliae. So this technique was really discovered by accident kind of around the same time in Japan and the Netherlands in flooded fields, where they realized these conditions could lead to destruction of these long-lived spores and ridding them of these problems in their fields. And so there's a lot of interest now here in the United States in further developing this technology and adapting it for our cropping systems. And so there's a lot of research going on now in California and Florida in large-scale production systems, as well as smaller-scale production systems at various places around the U.S. So just to remind you all, or if you're new to this, really how ASD technology is suspected to work is that you add a very labile carbon substrate to the soil and then saturate it with water, and you cover the surface of the soil with an impermeable plastic layer. And so the aerobic organisms in soil quickly deplete all the oxygen, and then you create these very anaerobic soil conditions which are harmful to those spores and that pathogen, which is they are aerobic individuals. And so that can start to break down those spores. And then also at the same time, you get all these anaerobic organisms that start producing toxic compounds that are also harmful to those spores. And so you deplete the soil of them, and then you take off the tarp and you have a clean field, and good organisms can recolonize. And so just a few quick updates on our studies around ASD technology and mint. So some of our objectives in this trial are first just to determine whether ASD can be used successfully in our environment to control the Verticillium dahliae isolates that are specifically effective against mint plants. We also want to understand and determine whether they can work in the multiple soil types where mint is grown here in our region, so both mineral soils as well as muck or organic soils, and then identify alternative substrates that can be successful here in our cropping system. So traditionally in Japan and the Netherlands, people are using things like molasses because they have sugar beet industries, but we don't have that, so we need to find resources that we can use locally. And then lastly, I want to point out this last bullet point here in that this research, while we're hoping ASD will work, we realize it might not be economically feasible on a large scale. It might have more relevance to like smaller scale nurseries where you're producing your transplants and you want to have clean transplants, but we're also using this project really to just better understand mint production, what you're doing, where there are points we could help to reduce this pathogen, and really, from my standpoint as a soil microbiologist, to understand more how this organism is surviving and how we can disrupt that life cycle so that we can have longer term solutions. So I put this slide in here because I had a question last summer in talking to a mint grower about, why would this technique, work? Why are you adding all this carbon when, especially to these high organic matter soils that already have a lot of carbon in them? And I didn't really do a good job answering that question at that time, and I think probably other growers have that question too. So I just wanted to point out that, when we talk about organic matter and you get a soil test back from a commercial lab, that's total organic matter in soil. But really, when we look into soils and soil organic matter, there's multiple pools. And so you have this, very active pool, which is more plant residues and things coming in, which are fairly easily broken down. Then you have longer pools, kind of slower pools that have already been processed by microbes. Those can last hundreds of years. And then we have really passive organic matter that has been through microbial decomposition, and it can last, for thousands of years potentially in soil. And so when we look at agricultural systems, anytime we start plowing soil, this figure here on the right shows us this is time since the start of cultivation, and these are the different organic matter pools. You can see that, once you start working the soil, those more active, kind of passive or slow organic matter pools break down over time. And what you're left with after many years is often that very more recalcitrant pool. And so while you can do a soil test and think you have a lot of carbon, you may, but you might not have the type that actually really supports microbes. And so it's really that very labile-like sugar that we're interested in that supports the most active anaerobic organisms. And so even in a very high organic matter soil, this technique still has capacity to potentially work in the way that we think it can. So the substrates that we're evaluating, first of all, we're evaluating a brassica cover crop. It's a mixture of different mustard species, including Brassica gentiae and Sinapis alba, which are already known for their potential to have some kind of biofumigant potential. And that's because these mustards produce glucosinolate compounds that, as they break down, they release compounds called isothiocyanates, which act much like chemical fumigants in soil. And so by using a brassica cover crop, maybe we can get kind of a double whammy in that biofumigant, but also the ASD kind of benefit. So we're also working with a chicken litter. So here in our region, we have lots of chickens, lots of turkeys, and they produce a lot of waste and bedding. And so there's a whole industry around taking those resources, heat treating them to kill the pathogen, and prelling them down as a nice soil amendment and actually fertilizer amendment. Next, of course, this is corn country, so we have lots of ethanol, but we realized very quickly, so people are trying ethanol in other places in ASD technologies, but it's challenging to get that out into the field, and it's actually a pretty valuable resource. So we're only evaluating that at this point in the greenhouse, and as I'll show you, it hasn't been that effective in the greenhouse, so we did not include this one in our field trials. Then we're using a dried distiller's grain, which is the kind of cake from the corn that's left over after the corn is crushed for ethanol production. And then also a soybean seed meal, which is the soybean cake that's left over after extraction for biodiesel. And so we have a lot of these resources here in the Midwest right now. They're primarily being used in animal feed, but we also think we can use them to feed the soil livestock through the ASD technique. And they have a lot of nitrogen in them as well. So with all of these cases, in addition to meeting the ASD technology, there's also a fertility benefit. So I'm not going to spend a lot of time on this, but these are some of the greenhouse trials that Sahar Abdelrazak, my former postdoc, was conducting, where we collected soils, one from a mineral farm, one from a mineral soil from a farm, and a muck soil from a farm. We obtained a very virulent, highly pathogenic strain of Verticillium on mint. We grew that, we added it to the soils, so they would be very highly infected. Then we added the substrates, filled them up with water like this, and let that kind of anaerobic process happen. And then we put those in pots and put mint transplants in. And so this is at harvest. You can see the little mint transplants after they've grown and the different treatments, where here with this little blue tag, you have a very healthy transplant, and over here you have some transplants that aren't working so well. And this is kind of the rating scale that we used, where 0, very healthy plant, lots of roots, lots of leaves over here, very clearly showing Verticillium wilt treatments. And you can't see it because of my picture, but there are no roots on this plant. So just quickly, some of her results. I know she talked about this last year, but for those of you who missed it, I just wanted to show these again. These two figures, these are in the two soils, shady being the muck and fair oil oaks being the mineral. What happened in terms of infectivity. And so in both figures, on the far right is the untreated control, where the pathogen survived very well and went on to infect the plants. And then these are the other treatments that we tried. And so the yellow kind of squares are around 2 treatments that actually worked well in both soils, that being the chicken litter and the mustard cover crop in the ASD treatment. And so that was pretty exciting. The DDGS worked well at shady, but not so much in the fair oaks, which we're not sure yet, but we need to figure out why. And then, as I said, the ethanol did not work well at all. So moving on, in terms of field trials, we were delayed in getting our field trials going because we had some trouble in 2019 getting our cover crops established, and 2020 was also a challenge due to COVID, but we managed to get 3 field trials going, and so these are just some pictures of that. This is Petrus literally paving the way through the field, get back to one of our plots. This is Pat, the graduate student who's now working on the project, collecting some soil samples. And then these are, you can see us setting up, these are 2 locations. This is the the brassica cover crop, and then you can see like back in here, this I think is the soybean meal, or maybe this is DGG, this is the soybean meal. Then we saturated them with water, covered them with the tarps, and then here's Petrus and Pat installing the sensors that allowed us to track the temperature under those tarps as well as the anaerobic conditions. So here's some pictures of a month later when we came back to remove the tarps. And so just a couple of things to point out. Here, this is in one of the, I think this was just the solarization treatment, no ASD, just the plastic cover. It kind of worked like a nice little greenhouse, stimulating weed seed development, which then went on to die before winter. So just to highlight, a lot of these strategies we think can be very effective, not just in killing the pathogen, but also weed seeds, which is always valuable. Then these 2 figures here, you can see this kind of stuff growing on the soil. That's actually fungus, good fungi that are breaking down some of those substrates. Here's a place where the cover crop was regrowing, so we're getting 2 seasons of kind of kill in, which was nice, and then collecting soil samples again. So what's next? We'll be coming back this spring, hopefully in late March, to transplant mint transplants into the field trials, and then we'll be also collecting more soil, looking at soil health and pathogen survival, evaluating the plots for disease incidence, and looking at yield and essential oil production. And then we do have a field trial or a field day set at the farm near Fair Oaks on June 22nd. So hopefully you'll be able to join us. So another thing we really want to do this summer that we had planned to do, we're a little bit behind because of some things, but we want to get out this summer. We think it's going to be possible. We have a team, including myself, Pat, the graduate students, and then 2 interns, Natalia and Jaime. And we want to come out and we want to get soil samples from all the mint farms around Indiana. So not just the ones where we're doing the on-farm trials, but all farms. And so the reason for that is we want to, again, getting back to that ecology and really understanding what makes this pathogen survive in one soil versus another. So we know, in some places it survives really well, in other places it might not. And so understanding what conditions, what soil conditions, switches soil chemical properties and soil microbial properties to allow that pathogen to survive and be successful or to be suppressed. And so that's where we really need your help in allowing us to come in and collect some soil samples to help us learn more about how to manage this pathogen. And so we'll be very happy to share the results with you of your soils and where they're at. So lastly, I just want to talk about a few things, kind of what we're learning, again, more about the ecology of this pathogen that might help us in coming up with new kind of control strategies in the future. And this, you can't see it here, but it says we want your feedback on this. So we want to hear the practicality of some of these things, and if you have ideas as well that we can look into and start to think about and come up with new collaborations moving forward. And so back to this cycle, we talked about how, the traditional wisdom is that these microsclerotia can be produced and they live in soil for forever, or like 14 years. But what we're learning is that they actually, a big part of their survival is potentially infecting other plants. And these strains that infect mint aren't going to show any disease symptoms on other crop species. They're asymptomatic, but it can be potentially a very important mechanism that allows the spores to survive over time or to be reduced over time. And so that gets into our evolving understanding about microbes and how they interact with plants. And so in the last 10 years, we have new molecular tools that are allowing us to learn a lot more about microbes in soil and microbes that interact with plants. And so we now know that, in addition to the pathogens like Verticillium that infect plants, plants are also hosting a whole bunch of other microorganisms called endophytes, which live inside their root systems. And so just classically, these haven't been well studied because they don't really display any visible symptoms. And so we're just learning about their ecology and how to leverage them to promote beneficial agricultural services. And so, part of this evolving understanding is realizing now that Verticillium dahliae isolates that maybe cause disease on mint can also infect other plants. Again, not showing symptoms, but this can be very important in their life cycle. And so some research out in the Northwest has shown that these isolates can infect alfalfa, red clover, barley, mustards. And so, while they don't cause disease in those plants, they are living in the root systems, reproducing, producing more microsclerotia. And this is really problematic, particularly around mustards, because they're seeing that the isolates are kind of evolving in the soil and adapting to those cropping systems. And so a lot of farmers in the Northwest are growing mustard cover crops as biofumigants to help reduce some of these disease cycles with things like Verticillium and pathogenic nematodes. But isolates that can kind of use mustard to hopscotch onto the crops that they're really interested in, again, are evolving, and so that's reducing the capacity of these crops to really have any value, these cover crops, in this system. So another thing, we're learning, again, back to these endophytes, and that while some plants can serve as kind of a hopscotch of these pathogens over time, there can also be a lot of good organisms that are living inside plant roots that can fight pathogens using various mechanisms, such as producing antibiotics that directly kill them. Much like when we're sick and we take antibiotics and it kills the pathogens, the same thing can be happening in a plant. And we know that how we manage most of these endophytes come from the soil, and how we manage soils can influence the composition of these beneficial microbes in soil that can go on to infect plants and help them fight disease. And so this is just an example from some of the work in my lab in carrots that Sahar Abdelrazak did during her PhD program, just in carrots, showing, again, all these endophytes that we isolated from carrot plants and evaluated their capacity to suppress a problematic pathogen there, Alternaria dahliae. And we found that we were comparing conventional and organic soils, but really, we think the main thing, the organic soils were healthier and supporting more of these beneficial microbes that could suppress the pathogen, because we had cover crops and things that were building the health of the soil and increasing the diversity and abundance of these beneficial microbes in soil. So this is something potentially we can look at in trying to leverage in mint production systems as we learn more about things that shift these populations of good and bad microorganisms in mint soils. And so, we can use this both, in mint production systems, but we can also leverage this in mint nurseries potentially. And this is where we think the ASD technology could be really helpful right now, while it might not be applicable at this point until we have large-scale equipment to do it in the Midwest. In bulk fields, we could use it in nurseries potentially to clean up the soil, seed the soil with beneficials, and then bring in, mint plants that not only, you're bringing in your transplant, but you're bringing in good organisms that are inside of those mint plants and can help fight any pathogens that might be present in the bulk fields. So another thing, that's our kind of evolving understanding of Verticillium is how these spores survive in soil. And so this was a study that came out recently looking at melanin production. So when you grow Verticillium in a plate like this, you'll see it's really dark, and this is when it starts producing those microsclerotia, and it has a lot of this melanin compound in it. Which is in a lot of organisms, but it happens to be a very characteristic in Verticillium. And so they did some studies, knocking out the ability of this microbe to produce melanin, and they discovered that melanin is not essential to produce these microsclerotia, but it does have a big effect on how long those microsclerotia can survive in soil and how susceptible they are to many stresses. And so in addition, they found that the type of crop or plant species that they're growing in, whether it's something that they cause disease in, or some of these different hosts where they're just asymptomatic endophytes, can influence their ability to produce melanin in their spores. And so this could be very big in terms of identifying crops that maybe they infect them, but they don't produce really viable microsclerotia. So also in this study, which comes back to the healthy soil, they found that having these antagonistic beneficial microbes around also reduced the capacity of Verticillium to produce spores that had this melanin that helped them survive in soil. And so that kind of comes back again to better understanding our production systems here in Indiana. Most of the research at this point looking at alternative hosts has been in the Pacific Northwest with, cropping systems that they're using that aren't really relevant here in the Midwest. So we need to be looking more at our hosts here of different corn, soybeans, maybe wheat, and not just the crop itself, but even the varieties. So a lot of my work in looking at varietal differences in plant microbe interactions, we see differences. And so I have worked with wheat before and different wheat genotypes and how they can make soil more disease suppressive or not. So I think there's a lot we could look at in terms of crop rotation and things, and better understanding ways to diversify our cropping systems and reduce the survival and maybe even eliminate it over time in our mint production systems. So that's my last slide. I thank you for your attention again and for supporting our efforts in this project. It's been really fun to learn about mint and this new soil-borne pathogen. Again, we'd really like your feedback, ideas, what maybe you've learned about Verticillium that might help us in our trials, what you think about some of these kind of alternative approaches so that we can work together in the future. And then again, to remind you that we really want your soil this summer as part of our efforts to learn more about the pathogen and how to control it. So we'll probably be reaching out to you at some point in the spring, but you can also reach me at my office number or my email account at Purdue. So again, thank you for your time.